Welcome to the Bodhi Academy. My name is Shrutika and today let us continue with our lesson from class level, cell cycle and cell division. So in our part 2 we have studied what is cell division and how cell division takes place in the cell, that is through the M phase, that is mitosis. So today we will study another type of cell division, that is through meiosis. So meiosis. So meiosis, the cell division that reduces the number of chromosomes into half and results in production of haploid daughter cells is called meiosis. So it helps in production of haploid phase in life cycle of sexually reproducing organism. It involves the following event. So meiosis is basically here what happens during cell division. The number of chromosomes that are present also divides and reduces to half and forms what? Haploid daughter cells. Okay, so you get cells with haploid daughter cells. So meiosis generally takes place in the reproductive cells. So meiosis is basically occurs in two phases. That is, first is the meiosis 1 and meiosis 2. So meiosis 1 takes place into 4 phases. That is, first phase is prophase 1, second is metaphase 2, third is anaphase 3, and fourth is telophase 1. Similarly, in meiosis 2, there is first phase, that is prophase 2, then comes your metaphase 2, then anaphase 2 and the telophase 2. So meiosis in cell takes place into two separate parts, that is meiosis 1 and meiosis 2. And meiosis 1 will undergo prophase 1, metaphase 1, anaphase 1 and telophase 1, and meiosis 2 will undergo prophase 2, metaphase 2, anaphase 2 and telophase 2. This is how meiosis takes place in the cells, that is meiosis 1 and meiosis 2. So now let us understand each of this in detail. So first we will see how meiosis 1 happens. So first phase in the meiosis 1 is the prophase 1. So prophase 1 will undergo different stages, that is during the first stage in prophase is leptotene. So in the leptotene phase, that is, this is the phases of prophase 1, that is leptotene. So in the leptotene phase, the chromosome becomes distinct and visible under microscope. Compaction of chromosome continues throughout the leptotene phase. Then comes your second phase of prophase, that is zygotene stage. So during this stage, what happens is chromosome starts pairing together, and the paired chromosomes are called homologous chromosomes. So homologous chromosome formation takes place in the zygotene stage of prophase 1. So after that, it undergoes the third phase, that is the pectine stage. During this, the crossing over between two sister chromatids of homologous chromosomes occurs for exchange of genetic material. So during this phase, the non-sister chromosomes which are there, they will cross over each other so that the exchange of genetic material takes place during this phase. Then comes your diplotene phase. This phase is recognized by dissolution of symphytonymal complexes and tendency to separation of bivalent except at the site of crossing over. This forms an X-like structure called as the chiasmata. So during diplotene phase, chiasmata formation takes place. Dikinesis is marked by terminalization of chiasmata, and the nuclear membrane breaks and nucleolus disappears. So after the diplotene phase, dikinesis takes place, wherein the chiasmata which is formed during the diplotene phase will disappear, and the nuclear membrane breaks and the nucleolus also disappears. Next phase in the meiosis is the metaphase 1. So during metaphase 1, the bivalent chromosome align at the equatorial plate and microtubules from the opposite poles of the spindle get attached to the pair of homologous chromosomes. Then comes your third phase, that is anaphase 1. So during anaphase 1, the homologous chromosome separates but sister chromatids remains attached at the centromere. And during the fourth phase, that is telophase 1, the nuclear membrane and the nucleolus reappears and cytokinesis follows. That is, cell division will take place, that is, cytoplasm will divide in the telophase. This is called as dyad of the cells. The stage between the two meiotic division is called as the interkinesis, and it is short-lived, that follows prophase 2. So after this, the cell will undergo prophase 2, that is meiosis 2 division. So now let us see how the meiosis 1 takes place. That is, first is the prophase 1, that is, the chromosome will be condensed and it will be in the center. There the nuclear membrane will disappear. Then it will undergo metaphase 1, that is, the chromosomes will align themselves at the equatorial region. Then in the anaphase stage, the chromosome will move towards the poles. The chromosomes will move towards the poles. And then in the telophase, what happens is cytokinesis will take place, that is, the cytoplasm as the chromosome and the nuclear material, as the chromosome, that is the genetic material, are moving at the poles. What happens is the cell also starts separating. The cytoplasm of the cell also starts dividing, and then it forms two daughter cells. So from a single cell you get two daughter cells. After this stage, the cell will undergo the prophase 2 division. So this is the prophase 2 division. That is, these are the two cells which are formed from the meiosis 1, that is telophase 1 division. After telophase 1, those two cells will again undergo prophase 2, that is meiosis 2. So this is the starting of the meiosis 2. Now let us see what happens at the meiosis 2 stage. So in the meiosis 2 stage, it is initiated immediately after cytokinesis, before chromosome gets elongated. Okay, so first phase in meiosis 2 is the prophase. So in the prophase, the nuclear membrane disappears and the chromosome becomes compact. So again the nuclear membrane will disappear and chromosomes will become compact, and they will condense at the center. Then comes your second phase, that is metaphase 2 stage. The chromosome align at the equatorial region, and then attachment of the kinetochores of sister chromatids takes place. Then comes your third phase, that is anaphase 2. Here the start with the splitting of the chromosomes of each centromeres of each chromosomes to move towards the opposite poles. And then the telophase, wherein the telophase 2 stage, in which the two groups of chromosome gets enclosed by nuclear membrane followed by cytokinesis to form tetrad of the cell, that is 4 haploid daughter cells. So these were the two cells which we got from the meiosis 1. So the starting of the meiosis 2 takes place by the prophase 2 phase. So these two cells which are formed from the meiosis 1 will again undergo metaphase 2. That is, it will first in the prophase, the chromosome will be condensed, the nuclear membrane will disappear, then the chromosome will align themselves at the equatorial region. Then in the anaphase 2, the chromosome will move towards the pole, and then in the telophase, at the telophase 2 stage, what will happen is each cell will divide to form 4 daughter cells. That is, this cell will divide, there will be cytokinesis which will take place, and this will form one cell, and this will form one cell with the chromosome material, that is genetic material in it. So this will divide and this will divide, and you will get what? 1, 2, 3, 4 cells. from a single cell. First it will undergo meiosis 1 and it will form 2 cells. Then from those 2 cells it will undergo meiosis 2 and it will form what? 4 cells after cytokinesis in the telophase 2 phase. So first what we had was one cell which underwent what? Meiosis 1 and you what? 2 cells, daughter cells. After that these 2 daughter cells underwent meiosis 2 and they will form what? 1, 2, 3, 4 daughter cells. So 2 each. One cell will form these 2 cells and from this cell we will get these 2 cells. So this is how meiosis takes place in the cell, and this is how cell division takes place through meiosis. That is, first the cell will undergo meiosis 1 and then it will undergo meiosis 2 and it will form from one single cell it will form 2 daughter cells, and from those 2 daughter cells you will get 4 daughter cells. And this is how cell division will take place with the help of meiosis. So now let us see what are the significance of meiosis. So meiosis forms the gametes that are essential for sexual reproduction. Crossing over introduces new recombination of traits, helps in maintenance of chromosome number of sexually reproducing organism, because meiosis is what? Reduction division, wherein the chromosome numbers are reduced and provide evidences of basic relationship of the organisms. So these are the significance of meiosis. So in this video we have learned what is meiosis, what are the 2 types of meiosis which takes place in the cell division, that is meiosis 1 and meiosis 2, and we have seen how meiosis 1 and meiosis 2 takes place in cell and helps in the cell division. And we have also seen the significance of meiosis in this video. So thank you for watching today's video. Please don't forget to like, comment, share, subscribe Bodhi Academy.